Thank you for the invitation of having me here, Dr. Hartinger, talking to you all about oral cancer. My wife, the title of my presentation is Who's Protecting from Oral Cancer? My wife is an oncologist. She says, you're humanizing your cells, because it should be what is protecting you from oral cancer. Oral cancer is a very serious disease, and you can see in this image oral cancer, oral cell carcinoma in the heart palate of this patient. Oral cancer can be modeled in animals by using carcinogens, and these carcinogens can develop cancer, oral cancer in animal. You can see in this image, you can see a mouse with developing oral cancer. If we look at these cells or these lesions histologically, we see that the classical feature of humans and mouse are very similar. You can see these epithelial islands infiltrating the connective tissue. You have the classical feature, keratinization pearls, nuclei that are enlarged, that are different than normal cells. So the clinical and histological feature between human and mouse is very similar. So what prevents this from happening? How is the immune system geared up to prevent at the epithelial level when transformations occur? How the immune system prevents this occurrence? Human cells, they have evolved to display all their protein contact to the immune system. And when they display this protein content, they are geared to show what are all the proteins that are inside of them. And the immune system has evolved in the thymus not to recognize our own protein, but to recognize all other proteins. So mutations in cancer, DNA mutation, create new protein sequences. Those new protein sequences become neoantigens, or tumor neoantigens. And neo stands for new, and antigen stands for protein that induce an immune response, are able to induce an immune response. So tumor neoantigen within the epithelial layer, they are picked up by intraepithelial Langerhans cells, you can see here in blue. Those epithelial cells display those tumor antigen to T cell, in this case to CD8 T cells. And those CD8 T cells with co-stimulation, they become activated, they migrate, they proliferate. And when they encounter the tumor antigen again, those modified protein, they recognize the molecular shapes of those modified protein. When they find them again as cytotoxic T cells, cells that are able to kill the tumor cells, they induce tumor destruction. So that's how the system is controlled. Critical are these presenter molecules that present these modified molecular shape that the immune system is recognized to identify. So as molecular shape, we have the T cell receptor in blue, and these two presenter molecules called major histocompatibility complex molecule 1 and 2. In the molecular model, you can see that this presenter molecule holds a peptide, holds a modified peptide, a tumor antigen in its groove. And the T cell receptor can recognize the molecular shape of this particular modified protein. So the immune surveillance, how is the immune surveillance against those transformation, against these new proteins that are coming because of mutations in tumors? The immune surveillance is regulated by regulatory T cells, by Langerhans cells, which are in color in the next slide, helper T cells, and these three cell types regulate the transformation of effector cells from simple CD8 T cells that recognize modified protein into cells that are able to kill and destroy tumor cells. This image seems complex, but if you would look at it from the right, to your left, you have regulatory T cells in green that affect Langerhans cells, and the Langerhans cells in turn can turn those CD8 T cells into CTL, which are the cytotoxic T cell lymphocytes in red. Additionally, there are these T helper 1 cells, the orange cells. Those cells also aid this transformation or differentiation of cells from CD8 T cell into a cell that can kill the tumor-infected cell. So in our research, we engineered the removal of MHC class II on Langerhans cells. So the MHC class II interact with regulatory cells and with helper cells. So the loss of this particular presenter molecule prevents the display of the tumor antigen to these helper cells. And therefore, what we found is that the CD8 T cell, despite their increase in number, they are unable to become cytotoxic cells or cells that can kill the tumor cell. So in this image here, we have failure of immune surveillance. If you look at the red cross, the Langerhans cells cannot interact with the green cell, which is a regulatory cell. They cannot interact with the T helper 1 cell, which is an helper in transforming the cytotoxic cells, the CD8 T cell, into cytotoxic cells. And therefore, we have tumor growth. Therefore, this very complex mechanism, we are trying in our research, we're trying to dissect what are the mechanism why certain T cells that are capable, as a feature that they do have, are capable of killing, do not become killer cells. That's the focus of our research. And therefore, to answer the question of who is the protector against oral cancer, we have essentially regulatory T cell, the green cell, that affects the Langerhans cells in the center. And the Langerhans cells, in turn, helps the orange cells, which is a T helper 1 cells, to produce the cytokines that are shown there, interferon gamma, TNF alpha, to make the CD8 T cell that potentially is capable of becoming a cytotoxic T cell to actually become a cytotoxin and control the tumor. So this is a complex story. Try to make it as simple as possible. And if you have any question, I welcome any questions, and thank you for your attention.